Here's a short glimpse into the research that we were referring to at the beginning of the video. This 2004 study shows how a diet high in seed oils damages the mitochondria of animals. A brief episode of STZ-induced hyperglycemia produces cardiac abnormalities in rats fed a diet rich in omega-6 polyunsaturated fatty acids. Materials and methods. To investigate potential beneficial effects of omega-6 polyunsaturated fatty acids, PUFA, male Wistar rats, 220 to 240 grams, were first fed an N6 PUFA diet, supplemented with 20% sunflower oil. Energy contribution from fats in this diet was 40% of total energy intake, similar to human consumption. Feeding 20% sunflower oil successfully magnified the omega-6 PUFA content in the heart, mainly linoleic and arachidonic acid. In Figure 1a, you can see the big increase in omega-6 content due to the excess consumption of polyunsaturated plant fats. Cardiolipin and mitochondrial morphology. Cardiolipin is a mitochondrial membrane phospholipid that is essential for optimal functioning of oxidative phosphorylation enzyme complexes and ATP generation. Structural damage to the mitochondria is associated with derangements in fuel utilization. Disappearance of total cardiolipin resulted in loss of oxidative phosphorylation activity and could explain the drop in mitochondrial ATP synthesis. Total cardiolipin decreased almost sixfold after omega-6 PUFA feeding, with a significant drop of ATP only in PUFA diet hearts. In Figure 5a, we see the negative impact of PUFA feeding on the cardiolipin structure of the mitochondria. Glucose and palmitate oxidation. Inasmuch as condensed mitochondria suggest lack of energy production, glucose and palmitate oxidation in the isolated heart were studied in parallel with heart function. In the PUFA diet group, glucose oxidation dropped further compared with the ND and PC groups. In Figure 6a, we see the consequence of the destruction of the cardiolipin structure. The ability of the mitochondria to burn glucose as fuel has almost gone to zero. Furthermore, this tiny level of substrate oxidation stayed more or less the same, even when the heart had to burn significantly more fuel for higher physical loads, as seen in Figure 6b. Inasmuch as changes in cardiolipin have also been associated with mitochondrial structural alterations, we scrutinized mitochondrial morphology in all the above groups. Figure 5a depicts a mitochondrion with a double membrane and lamellar cristae, which are typical in NC, ND, and PC hearts, meaning all of the other study groups except the PUFA group. A novel observation in this study was abnormal condensed mitochondria, but only in the PD group, the polyunsaturated fats diet group. See Figure 5b. Figure 5. Biochemical and ultrastructural changes in cardiac mitochondria after PUFA feeding. A on the left and B at the right. Representative electron micrographs of myocytes, showing normal and condensed mitochondria respectively. Some condensed mitochondria were observed only in PUFA diet hearts. Summary. In animals, a diet high in polyunsaturated fatty acids, in this case sunflower oil, led to these issues: an increase in omega-6 content, a sharp decline of cardiolipin, a drastic reduction of glucose metabolism, caused by morphological and functional changes of the mitochondria. As Dr. Seyfried fans, you all know that this situation constitutes the precursor, maybe even the cause of cancer, as the inability to burn glucose for fuel is the hallmark of cancer, i.e. Warburg effect. Thus, avoiding omega-6 fats is essential to keep your mitochondria healthy and to prevent cancer.